It's a pleasure to be here, catch up with all of you, and present you a little bit of my research on brain monitoring. I've been getting a little bit cold, but I hope my voice will carry until the end. Imaging the brain and brain function and its alterations with pathologies carries a big challenge, which is related to all the different elements that come into play and interact with each other to keep this going effectively. And so, of course, we have the neurons and their association in very complex networks that carry the information that makes up cognitive activity with electrical interactions on a scale of milliseconds. But then, of course, they need to be supported by very diverse cells, which we call glial cells, and by a vasculature that needs to be very adaptive and that neurons can stimulate to increase perfusion locally to areas that increase their activity. And this on a scale of more like seconds. In order to capture these different elements, these different spatial and temporal scales, people have come up with a lot of very smart technologies to measure different aspects of brain function. And of course, two techniques that you are very familiar with are electroencephalography and functional MRI. So the former tracking neuronal activity more directly in terms of the electrical interactions. So with very good temporal specificity on the scale of milliseconds, while fMRI, in its most used variant, is typically tracking the adaptations of the vasculature, so the increases in blood flow and volume that come with neuronal activity on a scale of seconds, but with a spatial specificity that can be a lot more improved. So this has motivated the combination of these two techniques to try to make the most of each of them and overcome the limitations of each of them. And of course, you can do this separately, so you could eventually capture the same, record the same modalities on the same participants under the same experimental conditions. But you really need to go into simultaneous recordings in certain cases, so which I'm listing here. So if you need to capture spontaneous aspects of brain activity that you cannot reproducibly promote in separate sessions, things like resting state, activity, aspects related to sleep, variability across trials, pathologies like epilepsy, where you really don't control the activity externally. And of course, if you have a strong aspect of reducing intersession variability or the aspect, the influence of training or habituation across sessions. So that's why you'd like to do it simultaneously. And of course, something we became very interested in doing was to try to do it at 7 Tesla to benefit from the increased sensitivity that you can get in fMRI, which you can then flexibly trade, as you know, by increased temporal or spatial specificity. And on the latter, this goal of eventually trying to pinpoint activity at the level of different cortical layers, which can give you information in terms of the inputs and outputs to a certain cortical region because they are differentiated across layers, and eventually give you information about causality of directionality of interactions. Why would you not want to do that at 70? The reason is that when you combine EEG and fMRI, you create interactions between the two modalities that interfere with each other. So the fields, the B0 field and the B1 that MRI uses create perturbations and artifacts on the EEG, and vice versa, the presence of the conductive wires of EEG in there disrupt to some extent the fields, especially the B1 field that is used by MRI. So you raise questions in terms of safety for the participants and for the equipment, and artifacts on both modalities. And unfortunately, most of these aspects become increasingly problematic as you go to higher field strengths. And at the time that we started looking into this, which was here with CIBM, with the EPFL, and colleagues here from Geneva as well, the biggest bottleneck really was the EEG artifacts. So we really had very strong artifacts of several orders of magnitude larger than the activity of interest. And so we had to put a lot of attention into trying to improve that. Today, that problem is very extensively mitigated, I would say, and it really benefited from approaches that combined tailored optimizations on the hardware side for EEG, combined with tailored optimization improvements on the algorithm side, on the signal processing. And so two main pillars of this have been the use of better designed EEG setups that have more compact transmission with shorter wires and better shielded so that you reduce the interference of EEG, of electromagnetic induction effects on EEG from the MRI. And then the addition of sensors, additional sensors that are there to pick up similar artifacts from electromagnetic induction and not EEG. And then use these to try to estimate the artifacts on the real EEG sensors and remove them. With some parallels to what Thomas was presenting in terms of noise canceling and its parallels to audio noise canceling, adaptive noise canceling. So for that, you need the hardware, the sensors dedicated for this, and special algorithms that do adaptive denoising using this information. And today, this is available in products on the market, so you can get this out of the box. You don't have to go do your own modifications before you can use that reliably. And so this would be an example of how EEG correction works today. So it's essentially a sequence of several steps. We start with very noisy looking EEG that you then correct for the impact of gradient artifacts, so the MRI gradients. The impact of the ballistocardiogram artifact, so motion induced by the pulsatility of the heart. Corrections related to spontaneous motion of the individuals and other residuals. And then you end up in the bottom with an EEG that hopefully and often looks quite clean and very similar to what you'd have if you recorded outside the MRI. So here's an example where the participant closed their eyes at this point. So you see here quite nicely the increase in alpha, posterior alpha activity, which is expected when you close the eyes on EEG. Here's an example of the improvements related to that correction on visual evoked potentials. So just to see, starting from a situation that is quite noisy in terms of the average responses, letting us recover a large positive peak, dominant positive peak at 100 milliseconds from the stimulus, which is what is expected. A recovery of a topography that is more occipital and of clear, so each row would be one individual trial of a response. A recovery of a consistent response across trials. So the current consensus today is that this type of modifications are highly desirable for EEG fMRI at any field, and particularly as you go to higher fields. We then turned our attention to the other side, to the MRI quality. And here we had a bottleneck, which was the need to have some space around the head to be able to have the EEG wiring and let it come out and reach the amplifiers. So we were working with coils that were not the most effective that the state-of-the-art has today. So with less receive elements and larger receive elements farther from the head, that introduced some limitations in terms of what we could do in terms of acceleration of the imaging process. And so, although we were gaining sensitivity by going to higher field, we were losing with these limitations on the coil. And this is, of course, what you can get today with the more state-of-the-art coils, like the famous Nova coil, so with denser receive arrays closer to the head, letting you have both more sensitivity and more flexibility in terms of acceleration to reach, for instance, better trade-offs in terms of spatial and temporal sampling, and particularly going to sub-millimeter resolution, which is necessary if you want to start differentiating different layers of the cortex. So the problem here is that these arrays are really designed for optimal MRI performance, and they leave very little other space there for other things, in particular EEG. But about 5 years ago, a Dutch group, they came up with a smart, very pragmatic approach where they replaced a part of the EEG wiring with much flatter and thinner cables that could actually pass through that really narrow gap in the coil. So they chopped up the wires on both sides and soldered a more suitable type of cable in between. And with this, they could demonstrate this proof of concept and acquire images that had a lot higher quality and really with the flexibility that you can get with state-of-the-art 7T MRI. So we wanted to add this, explore this idea and combine it with other improvements we had been done on the EEG side to now try to get really a setup that could really get you optimal quality on both the EEG and the MR sides. So we explored a similar modification of the EEG cap that we did in-house. At the time, we were in touch with people from Brain Products, so the main provider of EEG equipment for MRI applications. And they were working on a version as well of their own, so you can see a little bit different in terms of the type of wiring that is used, which is much smaller and thinner, in fact, resulting in an implementation that, in my opinion, is much more scalable than ours was, and which indeed now they are making available for users, for the market. And so at the time, it became clear that it would be very interesting to test both in the study that we were already doing. So they lent us one of their prototypes and we tested it as well. That's how it looks like today on just scanning on a Phantom. So a relatively compact setup that goes through the Nova coil and digitizes EEG very close to the head. So we ran a pretty comprehensive study to assess the quality and the functional sensitivity of this new setup. We tested on Phantoms and on humans, 8 healthy humans, and investigated these aspects of safety, of data quality, and functional sensitivity. We acquired both separate EEG, separate MRI, and combined acquisitions to really look at the impact of each modality on the other when they're there and not there. With both caps and with different structural, functional imaging, and field mapping modalities to better understand the interaction mechanisms. And explored now protocols, so one with a little bit better spatiotemporal compromise and one really going into sub-millimeter resolution. And you can already see here that the impact of EEG on this was quite moderate, which was a very positive first observation. So as I mentioned, we acquired very different types of data from these participants and then looked at different brain regions with parcellations like in lobes or more functional parcellations like the Yeo type of atlas into networks, and looked essentially at the impact of EEG on MRI in terms of signal-to-noise ratio, functional sensitivity, aspects like connectivity, and so on. And we also confirmed that the quality of the EEG could be recovered with what we already had in terms of corrections. So just in terms of conclusions, we found no major safety issues or practical constraints. We found some mild penalties in MRI quality, especially in terms of SNR. But as you go really to the end metrics of functional sensitivity, the impairments seem to get quite variable to a point where the variability across sessions and across individuals actually is superior to the losses you might have from having the EEG in there. So it becomes quite positive in that case. I think I had an example here, yeah. In terms of the impact on pairwise connectivity, so a connectome obtained with pairwise connectivity across parcels of the Desikan-Kiliani atlas, which is quite often used for that. And you can see visually very similar connectomes between a situation where you just acquire fMRI or if you acquire EEG as well. And we could assess that there were no statistically significant losses across these pairs for this participant group. So if you're interested in this, we've comprehensively described that in work that is now published in Imaging Neuroscience. And the data set is available as well on Zenodo. So if you're not so interested in all this quality aspects, but you'd like to apply your methods of functional analysis, connectivity, and so on, the data is there pretty much ready for that use, so already pre-processed and corrected for these artifacts. So you can right away apply your analysis if you're interested. What we've been looking at on our site was just have some preliminary ideas in this data set of whether there is interesting relationships between EEG and fMRI, whether they reproduce what's in the literature, and what is the benefit we gain with going to much smaller voxels. So here we just did ICA the composition of the EEG from combined recordings, either with fMRI at 1.6 or 0.8 millimeters, and then choosing components that had a very pronounced alpha peak, which is the dominant activity of EEG at rest, awake rest. And one observation we can already see is that ICA actually gives quite reproducible components between the two acquisitions for pretty much all the participants we had. And then we picked some of these, so one more with a source that is more occipital, closer to visual cortex, and one that had a dipolar source, which is more parietal. And we looked at what sort of fluctuations they explain in the bold data. So here for 1.6 millimeter and 0.8 millimeter images. So you can see already, and it gets quite cool that I have to zoom in these figures to show them to the voxel level in slides. So you can see how the specificity increases already in the anatomy of this. So this is really the EPI image. It's not an anatomical. Of course, there are more artifacts in the higher resolution data, but we then can see how, for example, the relationship between occipital alpha and the visual cortex becomes better depicted at 0.8 millimeters. And in the case of the other source, how it identifies networks that are containing the default mode network and other attention areas as well, and how you become much better depicted as you go to higher resolutions. So this is just an illustration in a few patients, but we're looking now as well at the effects across cortical depth. So that is my colleague Jonathan, who's very actively working into that now, and some results are starting to come up. So he'll be presenting that in weeks to come. And then at the same time, I've been looking at other scales of brain activity that have a lot to do with the expertise at my center. So in particular, what happens when we go to much longer time scales of days and months and years. In particular, the motivations of clinicians asking us and often complaining that a patient has a certain pathology, is undergoing a certain therapy, you do extensive exams on site, but then they go home and it's unclear whether they're really undergoing the therapy, how is it working, is the trade-off between therapy and collateral effects really worth it? And much of this today is still done by self-reporting of the patients with the clinicians, which is very subjective. Then the challenge, of course, is how do we measure this with current technology? Of course, we can't, because it's bulky, it's expensive to use. It's mainly limited to labs and hospitals. So then comes the expertise of my center. So very quickly, we are, for those who don't know, the Swiss Center for Electronics and Microtechnology, which is a research and technology organization, non-profit, non-academic, but dedicated to research, with a main mission to help promote innovation, do technological innovation, and transfer it to improve, help the industry grow, and particularly the Swiss industry. But we collaborate with not just the industry, but academics and clinicians. And one of our areas is, of course, in medtech. That's where I'm involved. And we've had a strong focus on wearable sensors, building on this tradition of microelectronics over the last 20 plus years. And at our department, really the focus we try is to cover all the bricks of a device, from the electronics, the sensing technology, to the operation of the device, the flow of data to the cloud and other infrastructures, and then the analysis with algorithms to extract meaningful information, as well as some testing, but then leaving the larger scale testing typically to clinical partners and other academic partners. So we started very early looking at optical signals for cardiac pulsatility. So with work in heart rate already more than 20 years ago, before many of the smartwatches that you have now were even thought about. And then looked at other aspects as well, other information that is contained in these pulsatile signals related to the mechanics and the health of the vasculature itself. And in particular, containing information, for example, about blood pressure, that as a gold standard can only be captured with more occlusive methods. So we work on various areas of remote monitoring, including movement and activity, both in sports and your everyday ratios of activity and rest. Optics, as I already mentioned, and electrophysiology was something we had worked a lot on for the heart. And then about 5 years ago, we started going more and more into brain activity, and that was the first sort of bridging that we did into EEG, was bringing this expertise in electrocardiography and exploring compact EEG devices. So we've now, over the past 5 years, looked at very different, developed very different proof of concepts and prototypes. I will not go into all of them. We've also been benefiting and quite impressed about improvements in dry electrode technology, so the ability to get good conductive contact with the head, including regions with hair, with dry electrodes without gel. There's been quite some good improvements in that. This is an example from a Swiss company that we often collaborate with. And I wanted to go a little bit into some examples. So this is the case of sleep monitoring. So sleep disorders are very prevalent and very underdiagnosed because the people affected are not conscious about many of these problems. So one device we did, very simple, is just a single channel device monitoring on the forehead, which is a region that you often don't need to rest on. So made to be simple, comfortable, and quite easy to use. So you'd plug it to this docking station, and the device recharges and uploads data. So with as little user interaction as possible. We've done some initial internal validations where we saw the ability to track the cyclic patterns of sleep EEG. And some microstructure, sleep microstructure features such as sleep spindles, delta activity. There are classic hallmarks of different phases of sleep. Then the question, of course, is this going to work in the wild with your random patient and not someone with a PhD in neuroscience or neuroimaging or someone who's on track to get it? And so in this collaboration with the Sleep-Wake Epilepsy Center of the University Hospitals of Bern, they collected a cohort of 16 patients that had late onset epilepsy, so a first seizure above 50 years old. And the patients were given the device to use it by themselves at home for roughly 1 month. So it's really trying to see what sort of quality of data we could get from this. And then we wanted to just get a sort of semi-automatic approach to determine how much sleep EEG data we could get per night that was of acceptable quality. So with some criteria in terms of the amplitude of EEG, the energy in that in the 0 to 30 hertz range, and the absence of some patterns that we knew meant lost contact with the skin. And then we could make these charts, so looking at the amount of sleep EEG data that was of usable quality per night over the patients. We've now analyzed 10 of these 16 patients, sorry. And so many of them, one interesting observation is that there is no first night effect. So this is something that people who look at sleep often realize, is that when you put someone in the clinic to do a sleep scan or a sleep PSG, they're in a different environment with many wires. And so this first night is actually not representative of their usual sleep. Sometimes people do 2 sessions of this to accommodate for that, but it seems that in most patients, when you start at your home with a relatively simple device, there isn't even any perturbation on the first night. So we've now analyzed these 10 patients. We're working on the remaining ones. We see very good quality in at least 7 out of the 10, which I was very positively surprised about, with amounts of sleep EEG per night that are comparable to what we expect in terms of sleep duration. So I wasn't a huge believer in at-home EEG at this scale before, but I'm getting more convinced as I see this type of results. And really, with really good quality, so this is the time frequency representation of sleep. Really good quality in terms of alterations in the frequency profile that are cyclic and are representative of lighter and deeper sleep stages. So this is just one quick test with k-means clustering of these spectral profiles, so taking basically each of these columns and clustering them, and essentially you recover this representation of what is almost a hypnogram, so a cyclic classification of sleep. Of course, automatic methods to analyze this data are really important. We really need to move away from manual reviews by experts. Once you start getting these volumes of data from patients from every night recordings over several months. And so we've had to work on automatic ways of doing, for example, staging of sleep. And of course here, one of the limitations is that you have to work with quite limited data. So you have a new wearable, a new device, you start using it, but you don't have large data sets with gold standard labels. So one thing I will go over very quick that we've been working on is leveraging existing large-scale data sets from conventional polysomnography, and then adapting them as much as we can to better approximate wearable data. So one way is trying to get to positions that are as close as possible to our wearables. The other is adapting their sensitivity and particularly their gains across frequencies to better approximate our devices. Just to very quickly show here what you get in terms of accuracy. So as you try to approximate the wearable, the performance on the original data decreases, but the performance on the wearable data does increase. So it does suggest that these approaches are worthwhile to try to approximate our target device, even though they decrease performance on the data sets that we're training on. Okay. Now, moving beyond neuronal activity, this will be much more brief, and it's something that's really we're starting now, is looking at other aspects of brain health that have to do with the vasculature, and particularly with cerebrospinal fluid and its role in the waste clearance processes, so the clearance of metabolic waste in the brain, which is gaining a lot of interest now. So, and particularly with this glymphatic system. So the idea that CSF flows along the vessels in paravascular spaces, gets filtered basically through the parenchyma and carries metabolic waste products out of the parenchyma. Some of the substances that are known to be involved and cleared to some extent by this system are amyloid beta and tau and alpha-synuclein involved in Alzheimer's, Parkinson's. So the idea is that disruptions in this system may actually precede many of the biomarkers that we now have for these diseases and may allow much earlier detection. CSF flow seems to be promoted by the pulsatility of the heart, of the arteries, by respiration and other vasomotor activities. Now the idea is, the question is, can we track this in a compact way? There are some methods, there's still ongoing research on developing methods to measure this, and they're particularly relying on MRI or PET, so techniques that are heavy and lab-based. But there are some methods that have been proposed that are much more compact and can be made almost towards a wearable type of device. And one that we've been interested in is near-infrared spectroscopy. So it's looking at light as it's emitted into the tissues and then detected. What these researchers in Finland have seen is that light is not only affected in the near-infrared range, it's not only affected by hemoglobin, which has been used very extensively to look at oxygenation and hemodynamic responses, But if you go a little bit longer in the wavelength, you also start getting some significant absorption from water. And because we know that CSF is predominantly water, and at the larger, about 99% water, compared to blood that is at about 80%, if I'm not mistaken, we can now, if we can track all these substances with appropriate wavelengths, we can now start distinguishing this information from not just blood, but also CSF. Using models so about how light attenuates for different wavelengths and different substances. This is just a prototype that I convinced my colleagues to build, which has 3 wavelengths that are favorable to these tests. So I wanted to see if we could reproduce these observations with this technique. And indeed, we can. I will just jump a little bit forward to finish and show. So essentially what we see is that we can track total hemoglobin and total water, which includes plasma and CSF. And essentially, they show an anti-correlation over time. So as the volume of total hemoglobin increases, or the concentration of water decreases, and vice versa, across different time scales, like the cardiac pulsatility time scale and respiratory as well. And this reproduced observations from the Finnish group that they had made earlier. The idea is that this is related to what's called the Monro-Kelly doctrine, so the idea that the skull is a rigid compartment and that variations in blood lead to variations in plasma. They have to compensate each other to keep in this rigid volume. Why is that significant? Well, some people are now seeing that the coupling, the stability of this coupling between blood and CSF can be altered in pathologies like Alzheimer's. So it could give us a proxy and some starting information about alterations in these diseases, potentially related to the lymphatic system, and so on. So just to conclude, continued advances in imaging technologies continue to help us probe further into the brain. 70 MRI for very highly resolved hemodynamics, compact wearable sensors for longer-term at-home monitoring. Combined advances in both the hardware side and the signal analysis really allow us to make the best in many of these situations. And there's lots to explore in brain function and brain health. We're very happy to discuss any of these modalities or any ideas you might have about that. So thank you.